First of all, I'd like to thank the organisers for this superb meeting. It's always a delight to be in Norway, and a particular delight to be here in Toronto. Yesterday when I arrived, there was cloud in the sky, so I sent an email to Stig saying, where is the sunshine? And today we have sunshine, so, you know, I mean, that's the kind of organisation that we can all really appreciate. So what I'm going to do is talk about aging and give something of a kind of overview of how we understand the complexity of aging and to connect this with our aspirations for a virtual physiological human. Now, aging is an interesting subject to investigate because in a way it's incredibly familiar to all of us. When we're almost as young as the simulated baby that we saw in an earlier presentation, we very quickly get to process information that we receive about how old a person may be. So you can look at the two images here. These are two photographs of the same guy taken about 50 years apart. You instantly see that the man on the right is old. There are all sorts of superficial cues that come from the face, from the colour of the beard, from everything. But if we were able to do a kind of MRI scan, we would see that there are changes in brain structure, in bone structure. If we were to do cellular and molecular studies, we would see that these changes pervade the entire organism. So aging is, on the one hand, an incredibly complex and familiar process, and I think this presents us with distinctive challenges in trying to understand and embed our knowledge of aging in the way that we want to work. When Stig asked for the title, I gave him the one that was on the first slide, but in fact, actually, this is my real title, which is why I think the VPH will be no use at all to anyone if it doesn't also age, and I hope I'll be able to justify that to you as I go through the talk. This is a slide I'll come back to at the end of the talk, but I thought I would put it here because it gives, in some sense, the destination at which I want to arrive. The first thing to say, and this is really important, is that in today's world, particularly in high-income countries like Norway, the United States, Japan, etc., age is the single biggest risk factor for more than 90% of the medical conditions that demand national resource and demand research attention. And actually, it's astonishing that in spite of that fact, remarkably little effort is understanding what intrinsic aging brings to the pathogenesis of these conditions. What I'm going to show you is the complex multifactorial nature of the aging process. Something that will connect with a point that Dennis made in his wonderful talk earlier is about the intrinsic stochasticism of some of these processes. Also, there is this very interesting relationship between, sort of how the system has evolved. There is evidence for gene action. There are evolved regulatory processes, in spite of the fact that the primary driver of aging is due to a whole spectrum of random molecular damage. And I think one of the challenges is to understand this interrelationship between what it happens just as noise within the system, but also how the system has evolved over many billions of years to recognize, to deal with, and to respond to noise. So that's really, in a nutshell, the agenda that I have and the kind of conclusion that I want to reach. I think in this field, we're all familiar with arguments about whether we should adopt top-down approaches, bottom-up approaches, or middle-out approaches. But I think when we look at a complex phenomenon, and particularly when we look at something as complicated as aging, it's, as it were, blindingly obvious that we have to do all of them. We have to be highly reductionist because we need to understand how molecules interact as a part of the phenomenon that we're studying. But with aging in particular, but also with many other conditions, as you get down to the detail, as you focus on a particular mechanism, for example, it may be telomere maintenance or it may be mitochondrial function, you've got to get that molecular detail, but when you're focused on that level, you are not seeing the whole picture. So what we have to have in mind all the time is the capacity to zoom back out through the levels and put the story together. Sort of this is perhaps obvious, but I thought I would put this slide up here because it reflects the intrinsic tension in the way that we have to work. Now, I showed you in my title slide a picture of what aging does to a person. But actually the thing that aging does to people that we're most conscious of is rather depressingly, that as you get older you become increasingly likely to die. And what this graph shows is a profile of age-specific mortality for a typical human population. Note that the age-specific mortality on the vertical axis is shown on a logarithmic scale. The reason for that is that in a high-income country now, if I plotted it on an ordinary linear scale, you would see sort of nothing differentiating it from the horizontal axis until we get to age 40 or 50 or 60. But when we look at it this way, we can see that yes, early life is a time of somewhat increased vulnerability for all sorts of reasons that are pretty obvious. Mortality rates go down as we sort of grow up. They reach their minimum, surprisingly, around about the age of puberty. And then what is so striking about these data is that your risk of dying is going up from age 13, 14 onwards at pretty much an exponential rate. So for a human being, for every 8 additional years you live, your chance of dying doubles. You'll see that there's a kind of blip that appears around about the age 20, a higher blip for males than for females. Sadly, we know entirely what this is about. It's about accidents. It's about suicides as well. These are causes of death that are not so much related to intrinsic biology. But the really important thing is that the aging process is not something that begins at 40 or 50 or 60 when people start to become sort of visibly, demonstrably old. It's something that begins very early in life. There's some very interesting pathological studies on young people who have died of traumatic deaths show that even if you look at very fit young men in their early 20s, a large fraction of the population will show, for example, the beginnings of atherosclerosis within the arteries within their heart. So the changes that occur that contribute to aging are changes that begin in the body, probably in utero, but that begin to produce these kind of cumulative effects later. Genetics. Genetics has been discussed already, and I'll endorse the view that we heard from Dennis that sort of the gene-centered view of the universe does not capture the realities of aging. But nevertheless, it is interesting to ask what genetics brings to the story. And most of us have grown up with the idea that genetic longevity runs in families. A number of studies, mainly studies done in Scandinavian countries, where the registers of births and deaths have been particularly complete for a long time, confirm that yes, there is significant heritability of human longevity, and the best estimates that come out from these studies are that genes account for about 25% of what determines the length of a human life. Now, does this mean that genes in some way program the aging process? And the answer is most definitely no. And the reason really is simple. I'm going to provide a very sort of superficial summary of the arguments, but if you're interested in the detail of this, and it really, really matters, then do take a look at the paper that's referenced at the bottom. Now, the first reason, if you ask people, if you ask members of the public, sort of why aging occurs, they will say almost always that, well, aging is a biological necessity because we have to clear out of the population the old ones who have had their time. We have to make room for the next generation. And they will then go on to say, if you press them on mechanisms, well, of course, it's determined by our genomes. We have evolved programs to make us age and die. But in reality, if you look at natural populations, you will see that any sign of old age, of senescence, is extremely rare. Animals in nature die young because they're killed off by all the hazards of the environment. So what that means, quite simply, is that there's neither a biological need, nor is there an opportunity to evolve a program for aging. If aging is something that nature never normally sees, then you certainly, cannot evolve a hypothetical set of mechanisms that would kill animals that got too old. And of course there's no need, because the problem that most populations face is how to keep their numbers high. There is also a problem which relates to our understanding of the way natural selection works. And this is that natural selection works at the level of advantage to the individual. And so even if you set aside the previous objections and imagine that a program for aging had evolved, Then if you had a mutation that somehow broke the program, then the organism that did not age would, other things being equal, enjoy a selective advantage. So you would see freedom from aging spread throughout the population. And we simply do not see anything like that. And out of the millions and millions of, say, nematode worms that have been screened in screens that look for alterations in lifespan, we have never seen a mutant that escapes the aging process altogether. So we have to set aside the idea that aging itself is genetically programmed, and that sets us with the puzzle of understanding why it is so pervasive across the biological world. And in order to understand that, I'm going to sort of begin the story at the beginning. This is the beginning of a human life. This is a human egg. Just at the moment of fertilization, on the right-hand side, you can see the sperm that is about to complete the fertilization of the egg. And of course, in this context particularly, it's worth reminding ourselves of the old joke that life is a sexually transmitted condition with an invariably fatal outcome. And it's the fatal outcome that I want to talk about. So in order to introduce this, I want to go in a way back to the beginning, but now it's my own beginning in the study of aging, quite a long time ago. And this was based on looking at the way that the molecular machinery within cells replicates itself. Effectively, the cell is an entity that it contains its DNA, of course, but it also contains all of the other molecular machinery. So what you have is generation after generation of macromolecules that are sort of replicating, replacing themselves. And the fundamental question is, given this, if you were to be able to characterize the molecular integrity of the current generation of the macromolecules within a cell, and then to forecast what the next generation, sort of the next time unit into the future, or the next cell division into the future would be like, that would be the molecular integrity of generation T+1. Now, you can plot a simple two-dimensional diagram in which you have the line of equality as the 45-degree diagonal, and then anything that is above the line is where it's actually getting better from one generation to the next, and anything below the line is where it's getting worse. And at the time, building this on quite a lot of the information that was coming out from the discoveries about DNA replication, transcription, translation, I built a model that is still entirely valid today, although it's somewhat generic. And this shows that the relationship between the integrity of the macromolecules from generation T to generation T+1 takes the form of this kind of S-shaped curve, which will cross the line in a couple of places. Now, the upper point is a point of stability, because if through random fluctuation the cell should go below that, it will come back. The lower point of equality is a point of instability, because if you go below that, you will very quickly deteriorate and fall apart in what was termed an error catastrophe. But the important thing about developing this model was that the model showed that energy is critically important, because energy determines how accurately you build macromolecules in the first place, or how quickly you get rid of your mistakes. And either of those routes is the way to keep the system stable. So this provides a degree of freedom on which selection can operate. And you can imagine that if you wanted to save energy, you might trim things so the cell has only a minimal region of stability, which might be sufficient for what it needs in life, but you make thereby a very considerable energy saving. And I put this together at the time as a very young biologist, reading the works of sort of people beforehand, and in particular being intrigued by the ideas of August Weismann, whom we heard about earlier. And Weismann gave us one really important concept in biology, and this is the distinction between germline and soma. Now, that distinction is of fundamental importance in the study of aging, particularly human aging, because the simple fact is that each one of us in this room today is sitting here with our bodies of about 100 million cells. And just reflect on this. Each cell in your body here today could, if the records existed, trace its presence here today through an unbroken chain of cell division that goes back 3.5 billion or so years. So in a sense, every cell that makes up your body today is the product of an extraordinary feat of near immortality. And yet, and I hope this won't come as news to you, you're going to die. And the reason you're going to die is because your soma, the sort of major structures of your body, are mortal. And this is what Weissman focused on, was this paradox between being products of an immortal germline and yet having this intrinsic mortality. So how do we explain this? Well, one of the ways we can explain this is by thinking about physiology, and particularly about thinking about the energetics of resource allocation as it occurs within the physiology of the body. So the body has to acquire resources, generally nutrients, from the environment, and during the course of its lifetime it has to produce an output of progeny, and what Darwin's law of natural selection tells us is that those organisms that are most effective in capturing and transforming resources into progeny will be most numerous in future generations. But if we take the lid off the black box, we can see that the organism faces some pretty difficult allocation choices, in that there are many competing metabolic activities in which effort has to be invested: growth, repair and maintenance, storage, reproduction, and so on. And what we're learning from a lot of the work that's been done over the last decade, beautiful work on the determinants of resource allocation, the metabolic signaling pathways, insulin signaling, and so on, is that these are influenced by a set of genes that have evolved precisely to handle this kind of allocation of resource. And it turns out that it's thinking about resource allocation that gives us a key to understanding why aging occurs, which will then lead us into understanding how aging occurs. And now, remember what I said earlier about animals in nature dying young, and you ask yourself, if you were your genome rationalizing this issue, how much of your resource should you invest in the maintenance and repair of the somatic structures of your body? And the answer is, you should invest enough to keep the body in good shape for the sort of period of time that it's statistically likely still to be alive in your evolved wild environment in which there's lots of danger out there. But you simply do not want to over-invest in maintenance and repair, because that will steal resource that would be better used for growth and reproduction and the rest of it. And the outcome of that is that we have evolved limitations in somatic maintenance and repair, giving us effectively what I many years ago called the disposable soma. And actually there are lots of interesting examples of organisms that lack a clear soma germline distinction, such as Hydra, in which we find that the organism is effectively immortal because the germline permeates the entire structure of the body. So you can cut Hydra up into little pieces and each piece will live, and they've been tracked in labs over many years and shown not to undergo any intrinsic process of aging. Now, I'd love to talk more about the paradox of immortality of germline versus disposability of the soma, but there isn't time for that, so we can talk about it at dinner if I'm sitting near you. We can also use this same logic very easily to understand why different species come to have the different lifespans they do. Because imagine that we had a kind of a hypothetical species, a mouse, in which what I've just described has occurred. Now assume that that species evolves an adaptation such as flight. So the mouse evolves its forelimbs to be wings, hey, it's become a bat. And suddenly life is less dangerous because it can fly and it can forage over wider ranges, so it pushes up its survivorship in the wild. But now if it does nothing else, it pays a price because it's not investing in maintenance to correspond with its improved life expectation chances, so the benefit that comes from that adaptation will be wasted unless it also upregulates its investment in maintenance and repair. And by upregulating its investment in maintenance and repair, it will live longer. So this is the sort of prediction that you can go into the lab and test. It predicts that the genetic control of longevity will be affected by natural selection operating on all the vast array of literally hundreds of different maintenance and repair systems, things like the antioxidant defenses, things like DNA repair pathways, things like protein clearance pathways. And it predicts that, sort of when we're looking at aging, that there should be better maintenance and repair in germline than in soma. This is something that has been confirmed very emphatically within the last few years, looking at how when embryonic stem cells begin to undergo differentiation, within a period of 2 to 3 days, they comprehensively downregulate the whole array of cellular maintenance and repair systems. This confirms the idea that aging is caused primarily by damage, that longevity is regulated by resistance and repair, but it also points to the central problem that concerns us with the VPH and with understanding aging, that it's going to be inherently stochastic, it's going to be inherently complex, and there's going to be a lot of plasticity and trade-off. So the idea that longevity is associated with maintenance of repair is something that a graduate student of mine tested in the lab a few years ago by taking skin biopsies from a range of 8 different mammalian species having lifespans that were as short as rodents up to humans and cows and other animals, growing the cells in culture, subjecting the cells in culture to the variety of stresses that mimic the bad things that can happen in life, and showing for each of the different stressors, quite a range of stressors, that there was a very clear positive association between the intrinsic capacity of the cells to withstand stress and the longevity of the species from which the cells were grown. We also see in studies of model organisms like nematode worms that the extended lifespan mutants that have been characterized over the last 20 years, pretty much without exception, gain their longevity through upregulating maintenance and repair systems. But there's something also very important that we can deduce from these. This graph here shows ages at death in 2 different isogenic populations of nematode worms. The one on the left are the wild-type worms, the dark bars. The one on the right are the first of the extended lifespan mutants to be characterized. And you can see the difference in the means is down to this genetic change. But something that people had overlooked that is incredibly important, because they don't usually plot the data like this. They usually plot the data as survival curves. What they had overlooked was the extraordinary variability within each population. Now, you may say, well, that's just biological variation, but I would remind you that this is Caenorhabditis elegans we're talking about, an organism that was chosen for biological study because of the precise determinism of its biology. The adult worm contains 959 somatic cells, not a cell more, not a cell less. The developmental process is programmed with precision, and each worm within the population completes its development within a very narrow time window. They're isogenic. You can grow them in mixed liquid culture, so you've reduced environmental heterogeneity. So biology explains variation in terms of either genetic variation or environmental variation, neither of which applies in these cases. What applies here is something that Dennis referred to earlier, and that is the stochastic variation, the intrinsic noise within the system. And you can see how in the development of the worms it is not apparent, but in terms of their lifespan, their aging biology, it's enormous. So genetics is the difference in the means. Chance is what explains this huge dispersion within the populations. I mentioned earlier the heritability of lifespan, and over the last decade there has been an enormous amount of time and money invested in big genome-wide association studies to try to identify the genes that are involved. I've been a part of one of the biggest of these studies. We just published a paper earlier this year, and I have to say the results are incredibly disappointing, because what is becoming very apparent is that when we look at the genetics of aging and longevity, there is a very large number of genes individually of very small effect, and the statistical power of the techniques we have available is simply not up to picking them out. It's probably also because they're interconnected in ways that these methods of analysis have not yet allowed for. So again, it underlies the message that this is complex and there are multiple interacting factors. So what are the mechanisms that drive the aging process? We heard earlier about the central dogma of molecular biology, and I challenge the central dogma not so much from the point that it's not right. It is right in a sense, in the way that Newtonian mechanics remains right, even though we now know with everything that has become apparent later that it's more complicated than that. But in terms of the information systems within the cells that govern the replication of DNA, the transcription, translation of DNA, we now see that this has to be embedded within a much more complicated set of processes that are all part of the intracellular machinery for keeping the whole show on the road. And wherever damage can occur within these pathways, we have compelling evidence that it does occur. And this is the damage that contributes to aging. The mitochondria don't fit within the neat sort of schema of the left-hand side, although they are connected in very important ways, as I'll show you in a moment. But the mitochondria are important, number one, because that's where the energy comes from that powers everything else, and number two, because they're also the major intracellular source of reactive oxygen species which is an important contributor of damage. So, you know, during the 1990s and perhaps in the early years of this century, I had spent quite a lot of time building models that would explore the significance of interactions between all these different mechanisms, and this is simply to pay credit to some of the individuals with whom I've worked over that period. You know, sort of perhaps most recently, about 10 years ago, we got a grant to develop something that we call BASIS, the Biology of Aging E-Science Integration and Simulation System. We wrote a wonderfully ambitious grant. It had 3 sections. We were going to develop in the first part a model of the virtual aging cell. In the second part, we were going to develop the virtual aging tissue. And in the third part, we were going to develop a model of the virtual aging organism, which was not going to be a human, but it was going to be a nematode worm. Well, we achieved quite a lot towards our first goal, and I think we were very happy, as were our funders, with the progress. But we realized very quickly that the second and third goals were completely sort of beyond the capacity of the project at this duration. Although it is really important now, as we move forward with realistic plans for the VPH, to think how these should be brought in. I want to talk just a little bit now about, I think, what is a very important issue, and that is, given that this damage at a fine-grained molecular level compromises the integrity of everything, and I mean everything that the cell does, how does the organism handle the consequences of the progressive accumulation of damage that will occur pretty much from the moment of conception, or certainly from the moment of germline soma segregation in the early developing embryo? How does it deal with it? And one of the important questions is, how do cells deal with the damage that accumulate within themselves, within the context of a multicellular organism, where if a cell goes haywire, it can be lethal for the organism because it gives rise to the development of cancer. Well, we've known about one potent mechanism for quite a long time now, and this is apoptosis. And apoptosis, programmed cell death, cell suicide, call it what you will, is a way of getting rid of unwanted cells. They may be unwanted in development, they're part of the sculpting. They may be unwanted in the hematopoietic system, because you've got to have a means of getting rid of autoreactive cells in the immune system. But also, you want to get rid of cells that are damaged, so badly damaged that they pose an increased risk of malignancy. And with aging, we get more and more cells being damaged, so we see apoptosis coming into play more and more often within aging organs and tissues. And some years ago, in a study of apoptosis in tissue stem cells, we provided what I think then was the first evidence of intrinsic age-related deterioration in functionality of stem cells within the adult body, when we showed that stem cells in mouse intestines were twice as likely to initiate apoptosis in response to a very low dose of genotoxic stress as they were in old animals. And over the last decade, we've seen some beautiful transgenic animal models that have messed around with the triggers of apoptosis, mainly the p53 pathways. And this was designed to try to make cancer-proof animals. Well, if you make a cell in a transgenic animal more likely to undergo apoptosis in response to a low dose of stress, you can in fact generate a mouse which gets many fewer cancers statistically over its lifetime. So the mission was accomplished. But very interestingly, these animals aged faster. And the reason they aged faster is by making cells initiate apoptosis in response to damage. Given that damage pervades the organism, it was accelerating the age-related loss of tissue cellularity. So this then sort of leads to the conclusion that not all damaged cells need to be deleted. You need to get rid of the high-risk ones, but the other ones, it may be better to keep them around. So this then led us, and this is something that has become apparent really only over the last 3, 4, 5 years within our group, is that cell senescence is the alternative damage response. Now, cell senescence is something, for those of you who know this field, was first observed back in the early 1960s by Hayflick and Moorhead. And people used to think that this was just simply aging at the cellular level, that you grow cells in culture, they have a finite replicative capacity, they stop dividing, and they die. Now, we were able to show that when they hit this replicative limit, they don't die. You can keep them going in culture for years, literally. if you keep topping up the median and keep them clean. So they're not dead, but they've just stopped dividing. And we were interested to understand what was going on here. And again, we were interested in exploring heterogeneity that had been observed many years earlier in cell cultures in a really major paper in Science. You probably wouldn't get a paper like this published in Science these days. But it was where the authors had taken a single cell, a single human fibroblast, grown up a mass population, and then at various stages in the lifetime of the culture had subsampled individual cells from that clonally derived population and showed that at various time points, tremendous heterogeneity was apparent in the division potential of individual cells. And this tells us something important about the stochastic mechanisms that contribute to cellular senescence. So together with a mathematical modeler, Pete Sozu, we put together a model which incorporated the multiple mechanisms that were candidates to contribute to replicative senescence. And we showed that by combining these within a model that was parameterized according to the available data, we could replicate this kind of heterogeneity in ways that no other hypothesis could do. And then in the lab, João Passos came from Portugal to do a PhD with us, and he did a remarkable experimental study which confirmed that although everybody was thinking at that time about telomeres and telomere shortening as being what drove replicative senescence, João's work demonstrated that actually it was heterogeneity in the integrity of the mitochondrial population that was determining this enormous stochasticity. So this was the necessity of putting together multiple mechanisms, driving the heterogeneity, and really sort of shifting the causality goalposts in ways that made the issue more intriguing and more challenging. Now, with the power of confocal microscopy, you can begin now to really characterize senescent cells in the lab, and you can do live cell imaging studies. You can visualize the damage that you see within the cells, so that you can get a lot of extra data. So we were able, on the basis of this, by extending the mathematical models we'd done, by doing a lot of in vitro experiments, to come up with an interpretation that has proved to be pretty robust. And this is that the cells are actually equipped with an integrating sort of sweeping up mechanism that captures signals from the variety of different kinds of damage that arise within the cell. And if that cycle captures evidence of sufficient damage being present within the cell, and if it hasn't triggered apoptosis, what it will do is trigger the introduction of this state of irreversible cell cycle arrest that's replicative senescence. So this is just as important, a kind of evolved system for containing the damage that may contribute to age-related deterioration. It's going on in our tissues all the time, and collaborators at the Mayo Clinic showed recently that if you intervene by targeting senescent cells within aged tissues, you can produce therapeutic benefits in the tissues of mice. Now, this is also relevant because if you look at some of the models that have been developed over recent years to try to understand the consequences of exacerbating damage, and this is done, if you imagine there are all these different maintenance and repair systems, if you can create an animal in which you've broken one of these systems, you may be able to accelerate the accumulation of lesions that would arise from the failure of that or the impairment of that maintenance system. And this has been done to spectacular effect, particularly by Jan Hoijmaker's group at the University of Rotterdam, where they've generated transgenic animals deficient in individual DNA repair pathways, and they generate animals with a clearly accelerated aging phenotype. It was also done 10 years ago at the Karolinska Institute in Stockholm, where they generated mice that increased the burden of mitochondrial mutation by putting in a defective DNA polymerase gamma. And what we have here is accelerated animals. So what we're seeing here is that you can generate these kinds of models, but they're not models of the true aging process because they're amplifying just one type of damage. But what is curious is that the phenotypes, although they're amplifying different kinds of damage, the phenotypes are remarkably similar. And I think the explanation for that is what I showed you from our understanding of the integrative pathway that sweeps up damage of different kinds. So it doesn't matter that the triggering damage in one case is a DNA repair enzyme that's broken, in the other case it's more somatic mutations in the mitochondria. They're just invoking the same activation of the senescence pathway. So this is underlining the importance of this pathway, but also telling us that these processes are quite complicated when we try to unpick the levels. Now, with an eye on the time, I'm going to accelerate through this next part quite quickly. But I want to pay particular credit to my colleague shown in the photograph here, Darrell Shanley, who a couple of years ago took over from me as director of the Systems Biology of Aging Centre that we have in Newcastle. And we have a paper published just the week before last in PLOS Computational Biology that really pushes this understanding of the systems regulation of cellular senescence quite a lot further forward. And it brings in other pathways than those that we have previously considered. And it investigates where there might be targets for successful intervention. And since this is published and readily available, in the interest of time, I'm going to have to speed through this quite quickly. But what we wanted to do was to put together the most complete integrated model of the players that we could. So we built on work that we had done previously. This is work that was done the last couple of years on the insulin signaling, so AMP kinase mTOR signaling pathway, looking at the regulation of resource allocation. So we took that model, which is described in the papers referenced at the bottom. We also had a model which had been developed in another group at Stanford for the FOXO transcription factor. We took the model that we had described in our paper in Molecular Systems Biology 4 years ago, and we added to this a fresh model that represented the status of the mitochondrial population. So there's a lot of different elements coming in order to build an integrated dynamic model which captures all of these different elements. And the paper in PLOS Computational Biology shows how, with a lot of lab work, we were able to use in vitro time course data. We were here using the model of irradiation of the cells to initiate senescence. So this is fast tracking the damage that would otherwise occur during normal aging. And again, I won't go into the detail here, but this was a sort of completely symmetric collaboration between our colleagues in the lab and those who were building the models. And then there was a huge amount of parameter estimation, work on identifiability analysis. And once again, I'm really just putting this up to acknowledge that this is an important part of the work, and it took a great amount of effort. There had to be then the fitting of the model to the in vitro time course data, and so on and so on. So what we have at the end of this is a complete model, but nevertheless a simplified model, for irradiation-induced senescence that combines all these different elements. And this confirms the findings of earlier work, which is that it's not the dysregulation of a single player only that affects these changes, but it actually carries this further forward to investigate where you might conceivably now be able to target elements of the network to improve the development of senescence within the tissue. And I think I'm going to leave that complicated story at that point, because I just want, in the last minute or two, to touch on another sort of aspect of the work we've been doing that I think is going to be necessary for building an understanding of aging into the virtual physiological human. What I want to do, first of all, is just to say, with this understanding of how aging comes about, we see that at its core is driven by a build-up of random molecular damage that compromises organs and tissues and cellular functions. But what we're also learning from epidemiological observations is that it's not all intrinsic. We know that, for example, people who live their lives in conditions where they're exposed to unusually high levels of stress or to adverse environments, people living with great socioeconomic disadvantage, or people just eating the wrong kinds of foods, they don't just get fat and sick, they age faster, because what they're doing is accelerating the accumulation of damage that is intrinsic to the aging process. But on the other hand, all the people here, sort of healthy, active people who have this wonderful sort of nutritious and healthy lunch and dinner, are doing things to empower their cells' maintenance and repair systems and actually slowing the accumulation of damage. We're learning again about the role of inflammation and about anti-inflammatory processes. So what this tells us is that human aging is much more malleable than we used to think it was. It's driven by this core biology, but again, the virtual physiological human has got to allow the capacity to incorporate some of these extrinsic lifestyle factors in the way that the damage plays out in the body. And we've been, since 2006, running a study in Newcastle called the Newcastle 85+ study, where we began with a laughably simple design. And the design was that we got the funding and the ethical approval to approach everyone in Newcastle born in 1921 in the year 2006, so the year of their 85th birthday. And we'd spent 2 years doing pilot work to be able to get a high level of recruitment. And I'm incredibly proud in a way to say that we managed to recruit 75% of our target population, which, bearing in mind that many of these people are living in care homes, and we recruited people with dementia because it's unethical to exclude people with conditions like dementia from research simply because they've got the condition. If you know, and this is where the ethics come into it, if you know that they would have liked to have participated, so we rely on family members. So over this time we've visited them on 3 successive occasions and got further information on the 4th occasion. And we've got an absolute mass of data that we've only begun to analyse. These are areas in which we've published reports already. So there's a huge amount of data here that may one day, I hope, be able to play a part in contributing to populating some of the more human ends of the spectrum of building the virtual physiological human. So that was pretty much my last slide. This is where I began. I'm not going to take the time to reiterate the points here, but I hope you will at least feel somewhat persuaded that this captures the relevance for the VPH. And with that, I'll just put up my acknowledgement slide. A lot of people have been involved in this effort and a lot of funding. Thank you very much for your attention.
All right. Is this working?
Yep.
Fine. Thanks a lot for this very inspiring talk. We have time for maybe 2 short questions or 1 longer. Well, we already have 2. I think Jerome, you were first.
Okay, I will shout. So in relation to the statistics that you showed that where you showed that heredity was not largely correlated with longevity. So I'm wondering whether there exists some data that an existence study but by gender, because there are huge differences between males and females in relation to cell oxidation, reactive oxygen spaces, and senescence.
Yes, yeah. I mean, this is a hugely interesting area, the difference in life expectancy between men and women, and I've written about that separately. I didn't include it within this for obvious time reasons. It is interesting. The analysis of heritability takes account of gender, so sort of those statistics would not be affected. The question is, why do women live longer than men? Probably that's something to discuss over dinner. Just a very, very short answer. There are very good evolutionary reasons, and they relate to the difference in the biology of reproduction between males and females and humans and mammals. The pattern is similar in mammals. Basically, we age because the body is disposable. And the sad truth for the men in the audience is that the male body is a bit more disposable than the female body. And it's to do with the intimate relationship between the health of the female body and the nurturing in the womb of the baby and then the postnatal attention. But that's too brief to capture the essence. But there is a very clear prediction that differences in maintenance levels will exist between male and female cells. And this has been confirmed so far only in rodents. There is a remedy for men if you want to boost your maintenance and repair levels and increase your life expectancy, but it involves castration.
Okay, I think I was the second question.
Yes, just a question concerning heritability. I wasn't totally sure that I got you right on. I think you said that the contribution of genes to the statistics was about 0.22?
No, 2.5, 1 quarter.
Yeah, 1 quarter, but that's okay. That's fine. But it's 0.2 or 0.25. Doesn't matter to my question.
Yes.
That is the contribution of heritability, isn't it?
Yes.
With epigenetics now being something that we know can also be inherited, that doesn't necessarily mean a contribution to DNA.
No, it's broad sense heritability.
Yes,
so
indeed.
yeah. I mean, epigenetics comes into it in two different ways. Just very briefly to pick up on that. One, epigenetic modulation of the life history. Epigenetics may influence the way the organism adapts to its circumstances. The other thing that is important is that epigenetic tags get screwed up like everything else in cells as an intrinsic part of the aging process. So partly it connects with damage, partly it connects with regulation. It's quite a complicated story.
Okay, I'm afraid we have to stop here, so let's thank Tom again for this very nice talk.